In this video, we are going to talk about enzyme structure and catalysis. We're going to talk about the role of enzymes, enzyme structure and function. We're going to talk about how substrate interact with enzymes, and then we will talk about what happens to an enzyme after it does. Enzymes are workhorses. They catalyze just about all of the chemical reactions that happen inside cells. Sometimes we call enzymes biological catalysts, which means that they speed up certain reactions by lowering the amount of energy needed to get those reactions started. We call this energy barrier the activation energy for the reaction. In industrial chemistry, for example, activation energy often comes from an external source, such as heat. Raising the temperature increases the motion of the molecules. The rate at which they move and bounce into their neighbors increases the probability that those molecules will collide with enough kinetic energy to form or break a chemical bond. But cells can't encourage chemical reactions by just turning up the heat. Many cellular components are temperature sensitive, and they operate best at the range of temperatures typically found in living organisms. Proteins, for example, begin to unfold when heated. So, cells use enzymes to begin to bring reactants together and position them so that their reactive parts are close together, which encourages existing bonds to break and new ones to form. All of these molecular manipulations that we are talking about take place in the enzyme's active site, which is the area on the surface of the enzyme where this chemistry happens.
Enzymes are proteins that carry out chemical reactions. They have two special features. First, they are highly specific. Each enzyme acts only on select molecules called its substrates. Second, enzymes are catalysts. They greatly accelerate the rate of reactions they carry out, and they are unchanged by the reactions they carry out. Enzymes don't get used up by chemical reactions. They carry out the same reaction over and over as long as their substrate is available.
Like we said, enzymes are proteins, and enzymes have a three-dimensional structure that allows them to interact with other substances in very specific ways. As we can see from the GIF, substrate enter the active site of the enzyme where they undergo a chemical reaction. The interactions between the substrate and the active site are weak, non-covalent interactions. Once the chemical reaction occurs, the newly formed products leave the active site of the enzyme, and then the enzyme returns to its original shape, ready to catalyze a new set of substrates. If we look at the figure, we can see that the first step involved with enzyme catalysis is the binding of the substrate. In this case, the enzyme's name is hexokinase. Notice the ASE at the end of the word kinase. Hexokinase, or kinase, is an enzyme that phosphorylates a substrate. That means phosphate groups get added to one of the substrates. In this case, the phosphate group is coming from one ATP molecule, and that phosphate is being taken from one of the phosphates on the ATP molecule and given to a glucose. Therefore, both glucose and ATP are the substrates involved with this chemical reaction. ATP and glucose enter the active site of hexokinase. They undergo a chemical reaction where the phosphate group from ATP is donated to the glucose, and the two products leave the active site of the enzyme as ADP and glucose 6-phosphate. But what makes the active site so special? If we look at the polypeptide chain that's on the slide, this is an unfolded enzyme. It's a series of amino acids that have been peptide bonded together to form a chain. The squares in purple represent the amino acids that will eventually come together to form the active site of a folded enzyme. The enzyme's active site is actually extremely small compared to the enzyme itself. If we look at a folded enzyme, as shown on the slide, we see that the 4 purple amino acids all join at one location where they form the active site. The active site amino acids may be spaced far apart in the primary sequence of the enzyme, but when the protein is folded, they come together to form the active site. And enzymes are highly specific for both the substrate and the reaction that is catalyzed. If we look at the square in the center of the active site, that's the substrate. It binds to the active site of the folded enzyme. If we were to change any of the amino acids in purple so that they had different chemical properties, the substrate may not bind in the same way to the folded enzyme. And the enzyme would not be able to catalyze the chemical reaction it's supposed to catalyze.
Enzymes have a recessed pocket on their surface called the active site. The shape of this pocket precisely matches the shape of the substrate. Molecules that don't bind snugly in the active site don't react. The shape of the binding pocket is the key to enzyme specificity. This enzyme is chymotrypsin, which breaks down protein molecules in the stomach during digestion. Note how precisely the large side chain of a specific amino acid fits in the depression within the binding site. That precise fit positions specific atoms of the substrate for reaction with atoms from the enzyme.
The molecules you see on the slide represent the 20 essential amino acids that make up all proteins. Some amino acids have R groups that are non-polar. Some R groups are polar or positively charged or even negatively charged. The different amino acids that make up proteins influence the three-dimensional structure of the protein once it folds. The way substrate interacts with the active site of the amino acids determines the way the enzyme functions and the types of chemical reactions that the enzyme can do. If we look at the diagram shown here, we will see that the substrate in green interacts with different R groups from amino acids in the enzyme's active site. Serine, for example, is a polar amino acid. The polar R group of serine interacts with the OH group on the substrate's surface, forming a hydrogen bond. Similarly, lysine here at the bottom has a positively charged R group, which interacts with the negatively charged group on the substrate. And lastly, valine, alanine, leucine, and valine are all nonpolar, and they interact with this hydrophobic molecule on the surface of the substrate. Due to these non-covalent interactions between the substrate and the enzyme, we form the enzyme-substrate complex, which stabilizes the enzyme and the substrate to allow for a chemical reaction to take place. The figure on the slide should hopefully help you see how the substrate and enzyme interact. The substrate binds to the active site of the enzyme based on its chemical properties. The chemical groups on the substrate and the R groups on the amino acids of the enzyme bind together to form a stable complex. This is the enzyme-substrate complex. The model we see represented is called the Induced Fit Model. And this occurs when the enzyme changes shape slightly so that it surrounds the substrate and allows the substrate to change its shape. Based on that interaction, the chemical change occurs and the products are produced. Let's take a look at this practice question. A researcher proposes a model to explain how enzyme-substrate interactions determine enzyme specificity. The model is based on the idea that substrate molecules form favorable interactions with the amino acid side chains in an enzyme's active site. Based on the model, which of the following statements best explains an enzyme's specificity for a particular substrate molecule. Take a look at the answer choices, pause the video, and then come back when you're ready to review the answer. Okay, well, let's hope you chose letter D. D says a molecule with negative charges interacts with positively charged side chains in the enzyme's active site. Every enzyme has an active site. The active site is formed at the tertiary or quaternary level of protein structure, and it's where the chemical transformation of substrate to product occurs. Most enzymes act on a very specific preferred substrate based on the chemical interactions between the substrate and the amino acids of the active site. Although some will act on multiple substrates that are similar to each other. When the substrate enters the active site of the enzyme, the substrate and the enzyme bind together to form an enzyme-substrate complex. The enzyme and substrate interact through transient hydrogen bonding and ionic and hydrophobic interactions between the substrate and the R groups of the amino acids. These non-covalent bonds position the substrate in ways that favor a very specific chemical reaction to occur. The most important thing to remember from this slide is that an enzyme changes its shape, or conformation, as the substrates are converted into products. But the chemical reaction is complete. The enzyme will revert back to its original conformation, and it will be ready to receive another substrate molecule.
Enzymes work by lowering the activation energy of a reaction. Biological molecules are generally very stable. They don't spontaneously break down because the activation energy for that reaction is too high. Enzymes lower the activation energy by positioning the substrate in just the right shape at just the right distance from a reaction partner for the reaction to occur in the active site.
Chemical reactions involve the conversions of reactants to products. In a chemical reaction, some chemical bonds are broken and others are formed. In some chemical reactions, the chemical energy of the products is higher than the chemical energy of the reactants. And in other chemical reactions, the chemical energy of the reactants is higher than the chemical energy of the products. Regardless of the energy levels of the reactants and the products, all chemical reactions proceed through a transition state that has a large amount of energy. The transition state is an intermediate, highly unstable stage between reactants and products in which old bonds are breaking and new ones are forming. The difference in energy between the transition state and the reactants is called activation energy. It represents an energy barrier. All chemical reactions, whether they consume energy or release energy, require an initial input of energy to overcome this energy barrier. In chemical reactions that take place in a laboratory, this input of energy may take the form of heat. But in a cell, the energy barrier is reduced by enzymes. The rate of a chemical reaction is inversely related to the height of the energy barrier. The higher the energy barrier, the slower the reaction. And the lower the barrier, the faster the reaction. Enzymes, therefore, accelerate chemical reactions by reducing the activation energy of the reaction. This figure on the slide shows us the energy changes that would occur for a hypothetical exergonic reaction. An exergonic reaction is a reaction that involves the release of energy. In this case, there are two reactants that are swapping portions of their molecules. A, B is one of the reactant, and CD is a second reactant. The activation of the reactants is represented by the uphill portion of the graph, in which the free energy content of the reactant molecules increases. Once at the top of the hill, The activation energy has been absorbed by the molecules. The reactants are now in their transition state. They're activated, and their bonds are about to be broken. As the atoms then settle into their new, more stable bonding arrangements, energy is released to the surroundings. This corresponds to the downhill part of the curve, which shows the loss of free energy by the molecules. The products that form are A bonded to C and B bonded to D. As we said, chemical reactions require bonds to break and new bonds to form. For bonds to be broken, they must be strained or otherwise made less stable so that the breakage can occur. Getting reacting molecules into a less stable or more unstable state requires a small input of energy. Imagine a ball sitting at the top of a hill. For the ball to roll down the hill and release its stored energy, an initial energy must be applied to the ball so that it reaches over the small dip of the top of the hill. Once we get the ball over the barrier, the energy that was invested will be returned as the ball rolls down the hill spontaneously. In order for this reaction to proceed quickly, the barrier can be reduced. Enzymes speed up chemical reactions by lowering that barrier. This is the activation energy. In the case of our ball analogy, an enzyme would reduce the amount of the initial energy needed to push the ball down the hill. Once the enzyme has lowered the activation energy, then the rate of the reaction can be dramatically increased. Here's a practice question. A researcher proposes a model of an enzyme-catalyzed reaction in which a reactant is converted to a product. The model is based on the idea that the reactant passes through a transition state within the enzyme-substrate complex before the reactant is converted to the product. Which of the following statements best helps explain how the enzyme speeds up the reaction? So pause the video here, read over the answer choices, and return back when you're ready to review the answer. Okay, well, welcome back. I hope you chose letter B. The enzyme's active site binds to and stabilizes the transition state, which decreases the activation energy of the reaction. And the explanation here is that an enzyme is speeding up a reaction by stabilizing the transition state in the reaction. It decreases the reaction's activation energy because it binds more tightly to the substrate as that transition state is occurring. And this allows the reaction to proceed faster. We can think of the different steps involved with enzyme catalysis using these 5 steps. First, the substrate enter the active site of the enzyme. The substrate have a very specific shape that allows them to interact with the amino acids that make up the active site. Once the substrate have entered the active site, they'll be held in place via weak interactions and will form the enzyme-substrate complex. The substrates get converted to products, and the products get released from the active site of the enzyme. Once the products get released from the active site, the enzyme's active site is available for new substrates to bind. There's a 3-dimensional shape change that has to occur between step 3, 4, and 5.
When the reaction is completed, the products diffuse out of the enzyme. You might wonder why the products don't stick in the active site, since the substrate fits so well. It is because the products are now different molecules with bonds and electrons in different locations. The active site binds the substrate with just enough energy to promote the reaction, but the product diffuses freely away. When the reaction is completed, the enzyme is exactly as it was before the reaction, ready to react again.
Let's review another practice question. Which of the following statements best helps explain the reaction specificity of an enzyme? Read over the answer choices, pause the video, and then come back when you're ready to review the answer. All right, so welcome back. I hope you chose letter C, The shape and charge of the substrates are compatible with the active site of the enzyme. The justification for that answer is that the specificity of an enzyme is determined by the way it interacts with the substrates. The substrate shape and its charge affects how it fits into the enzyme's active site and whether it forms favorable interactions with the enzyme. One thing to think about with this question deals with the idea of mutations. If a mutation were to occur that changed the shape of the enzyme, that would change the way it catalyzes chemical reactions. Here's another practice question that we can review. The enzyme hexokinase catalyzes the conversion of glucose to glucose 6-phosphate, which is an important step in glycolysis. The reaction involves the transfer of a phosphate group from ATP to glucose. Either a glucose molecule or a water molecule can fit in the active site of hexokinase. The presence of a water molecule in hexokinase's active site would result in the hydrolysis of ATP to ADP instead of the conversion of glucose to glucose 6-phosphate. Which of the following statements best helps explain the reaction specificity of hexokinase? So look over the answer choices and see if you can choose the best answer. Then come back and we'll review. Okay, this time actually pause it. All right, fine. The answer is D. Glucose has the right shape and charge to cause hexokinase to undergo a structural change needed for catalysis, whereas water does not. All right, so that concludes our discussion of enzyme structure and catalysis. I'd like for you to review these questions and make sure that you understand the content involved with enzyme structure and catalysis. If you have any questions, please let me know.